One thing you must be extremely careful about in the ATP paper is avoiding the common examiner traps. Many students lose easy marks by making small mistakes rather than because they do not know the biology. Avoid mixing different units or forgetting to write units with your answers. Never draw tiny diagrams, add shading, or sketch with rough lines. Make sure you choose the correct graph, label every axis properly, and remember the bars should only touch in a histogram, not in a bar chart. During image comparison questions, compare only what you can actually see and never use outside biological knowledge. In planning questions, do not forget your control variables or the need to repeat the experiment. Instead of writing only, human error, as an improvement, suggest specific improvements. Measure the correct part of the specimen, never ignore a scale bar, and if the question asks you to draw only one part of a specimen, do not draw the entire specimen. Finally, here are the golden rules of ATP that can help you score the highest marks in the examination. Always read the question carefully twice before answering. Underline the command word first and answer only what is asked. Use a sharp HB pencil for all drawings and graphs. Make your biological drawings large enough to fill about 70% of the available space. Draw only what you can see and never add structures that are not visible. Use single, continuous outline lines without shading. Label diagrams neatly with straight ruled lines on one side and avoid crossing label lines. Measure the longest dimension of the specimen unless instructed otherwise. For magnification questions, always convert measurements into millimeters before calculating. If a scale bar is provided, measure the scale bar rather than the specimen. Show every step of your calculations and round your answers to one decimal place unless the question tells you otherwise. Also remember to always include units in your calculations, final answers, table headings, and graph axes. Choose the correct type of graph for the data provided. Whenever you describe a graph or table, support every statement with actual values from the data. In planning questions, clearly identify the independent, dependent, and control variables. Keep your methods clear, specific, and easy to repeat. Improve reliability by repeating the experiment and calculating the mean. When suggesting improvements, mention specific equipment or procedures such as a colorimeter, digital balance, larger sample size, or more repeats instead of giving vague answers. Always include suitable safety precautions whenever chemicals, heat, or sharp instruments are involved. Base every observation only on the information provided in the question and never make assumptions using textbook knowledge. Most importantly, think like a scientist by observing carefully, measuring accurately, and supporting every conclusion with evidence. And most importantly, practice as many ATP past papers as you can, because regular practice is the key to developing the observation, drawing, calculation, and data analysis skills needed to score well in this paper. So, that's all for the ATP. So, with that, we have officially completed the entire O-Level Biology 5090 syllabus, including the alternative to practical paper. You can find the PDF notes for ATP preparation through the link below. I wish you all the very best for your CAIEs. Thank you so much for watching. Goodbye, and all the very best.